Hello, welcome to Anatomy and Physiology 1. This is lecture number 2, the cell structure. This is part 1 of 2. Disclaimer, welcome to Anatomy and Physiology 1, lecture 2. I have put together a series of A&P 1 presentations that will be posted at later dates. I hope this will help A&P 1 students. Please take your time while reviewing this lecture. You may use your textbook to cross-reference. To have a clear understanding of this subject, I suggest you review this presentation before and after you have done your reading. Do not use this presentation as your only source of learning. Listen to your teacher/instructor, read your textbooks, and take notes. This slide is for entertainment and educational reference only. I am not a teacher. Please see your teacher/instructor for any questions that have not been addressed here. The cell structure. All organisms are composed of cells. Cells are responsible for everything that is living, whether it's one cell to a million cells, to trillions of cells. It is important to understand the cell because it directly affects how the human body works, how diseases works, and the rationale for therapy, whether it be medication, physical therapy, occupational therapy, all of the above. Concept of a cellular structure. After you have listened to this presentation, you've listened to your lecture, you've done your reading, there are two things that you should be able to do. The first thing is outline the major components of a cell. The second thing would be to discuss the developments in microscopy that have changed the view of the cell structure. Basic components of a cell. So first, before we go into basic components of a cell, let's define a cell. Let's talk about the cell. The cell is the smallest unit that can live on its own and that makes up all living organisms and the tissues of the body. The cell has three main parts. So, the cell has a plasma membrane, which is the borderline. You see this borderline here in the picture? Okay, and it defines the boundaries, okay? It's what gives it its shape. Then you have the nucleus, which is this purple here, just the outside, the circular. But the way the diagram is made is so that you can see what's inside of the nucleus. But we'll focus on what's only the nucleus at this point. Okay, so that's the nucleus. And also we have the cytoplasm, which is a gelatinous liquid. It is 80% water. It contains dissolved carbohydrates, proteins, and fats. It is also composed of water, salts, and various organic molecules. Intracellular organelles, such as the nucleus and the mitochondria, they are enclosed by membranes that separate them from the cytoplasm. The cytoplasm is also a medium for chemical reactions. It performs a platform for organelles to operate within the cell. It allows the cell to expand, to grow, and to replicate. Cytoplasm is external to nuclear membrane and internal to cellular membrane. Remember I told you that this cytoplasm is a gelatinous liquid? Within that gelatinous substance, the cytoplasm, is another intracellular fluid, also known as ICF. It's called cytosol. Cytosol is just the liquid, and it is made up mainly of protein, and it does not contain any organelles. Now, outside of the cell, fluids that are outside of the cell itself is called extracellular fluid, ECF. Extracellular fluid includes interstitial fluid. Now, interstitial fluid is the fluid that is found outside of the cell where the cells are, and they're mainly near blood vessels. It's fluid where the cells move around in. The plasma membrane. If you look at the picture in the frame here, you see that it is part of a cell. It shows you the nucleus, the nuclear envelope, plasma membrane of the lower cell, what else? Intercellular space, and plasma membrane of the upper cell. And this is the membrane here. Remember I told you that it keeps the border. It's actually the border. So it gives it its shape. Okay? And this is what the cell looks like through an electron microscope. Well, what does the plasma membrane do? It defines the cell borders. It governs the interaction with the other cells and controls passage of materials in and out of the cell. Membrane lipids. 98% of membrane molecules are lipids. There are three main types regarding the membrane lipids of a cell. Okay? Let's discuss the first one. So phospholipids, 75% of the membrane lipids are phospholipids. There are three different types depending on how they are positioned within the membrane wall. There are amphipathic lipids, which are molecules arranged in a bilayer. There are hydrophilic, which are phosphate heads that face the water on each side of the membrane. Then there's hydrophobic lipids, which the tail are directed towards the center, avoiding water. Doesn't like water, doesn't want to go near the water. Okay, they drift laterally, keeping the membrane fluid. Then there's cholesterol. They are 20% of the membrane lipid. They hold phospholipids still and can stiffen the membrane, so it gives it that solid shape. Then there's glycolipid, which are 5% of the membrane lipids. The phospholipids, which short carbohydrate chains on the extracellular face. The extracellular face would be the outside of the membrane, okay? Intercellular would be facing the inside of the membrane. And it contributes to the glycolytic carbohydrate coating on the cell surface. Membrane protein. Okay, there are 2% of the molecules, but 50% of the weight. Okay, there are integral protein, which I'm going to explain to you in a few minutes. And then there's peripheral protein. Okay, they adhere to the surface, to one face of the membrane, but they do not penetrate it. And usually they are anchored or tethered to the cytoskeleton. Okay, so let's discuss that. Well, first of all, here's your membrane lipid. This is the phosphate bilayer. Remember, here's one bilayer. Here's the other layer. So two of them equals the bilayer. Okay, then you have, these are proteins here. These are protein. This is the carbohydrate. This is the cytoskeleton protein right here. And this here is the anchoring peripheral protein. Now, let's talk about the protein bodies. Okay, the regions and so forth. Okay, this is called a transmembrane protein. The reason why it's a transmembrane protein is because here's the bilayer. It goes above the bilayer here and here on both ends. Now, of the transmembrane protein, you have the hydrophilic region. Remember that region, it likes to go towards the water. In the middle right here, that is the hydrophobic region. It's hydrophobic because it's in the middle and it really doesn't want to go towards the water. Okay, it doesn't want to get wet. Doesn't like that. Now, this is if you had a protein and it's actually embedded, from through it, like end to end, but that doesn't go out or over, that is called an integral protein. Now, here's the thing. An integral protein cannot be a transmembrane protein. However, a transmembrane protein can be an integral protein. Keep that in mind. Microvilli. Okay, so remember the cell where I showed you it had all the stuff inside of it? Okay, if you had looked to the very top, you would have seen some some spikes. Like, let me see if I can draw it for you. So you have the cell. Okay, excuse my drawing, I'm not an artist. Then you had this stuff here. Okay. You had the nucleus. And you had all the other stuff in there. All the other stuff. Remember, you had the mitochondria. The 1, 2, 3, they were in the corner, okay? These right here, that's the microvilli. What it does is it gives the cell more surface area. It gives the cell membrane more surface area. Okay, these are usually found, microvilli is usually found in cells that are specialized for absorption. You would find these mainly in the intestine when they absorb the nutrients. In some cells, some specialized cells, they are very dense. They appear like a brush, and they have cilia-like, very thin hair-like extensions. I call these little microvilli. That's the proper term, microvilli. Okay? Flagella. Flagella, the tail of a sperm, only functional flagellum in humans. So you have the sperm, and that when those little sperms are swimming and that little tail at the end, that is called a flagellum. Okay? They're whip-like structure with axomy identical to cilium. They're longer than cilia or cilium, and they're stiffened by coarse fibers that support the tail. Their movement is undulating, snake-like corkscrew. There's no power stroke or anything. They just move. They're just moving. Okay, that's for the flagella. You can find them in some bacteria, but it won't be in humans. Okay, but they do exist. Membrane transport. What does the cell membrane have to do with transporting? Well, after you have gone over this section, what should you know? Well, you should be able to explain what is meant by selectively permeable membrane. You should also be able to describe the various mechanisms for transporting material through the plasma membrane. Membrane transport. Plasma membranes, which is, remember, the border that keeps the cell, it gives it its shape and everything. Well, it's selectively permeable, meaning it only allows certain things to go in and out of it. It stops things from coming in. It allows things to go out. It allows things to come in, and vice versa. It just sometimes it'll stop it completely. Okay? Passive mechanisms require no ATP. That's something we'll go over later. Okay, so random molecules, motion of particles provide necessary energy. Then you have filtration, diffusion, and osmosis. Those are passive mechanisms that require no ATP. Then you have mechanisms that will need the ATP. ATP is actually energy. Okay, these active membranes consume the ATP. That is active transport and vesicular transport. Carrier-mediated mechanisms use membrane protein to transport substances across the membrane. Okay, so let's talk about the first mode of transport. The first mode is filtration. Filtration is allowing particles that are driven through the membrane by physical pressure. An example would be, forget about this for 1 minute, but you know when you make coffee and you have a filter and the water just runs through from the pressure of the water, it's similar to the same thing. With this, okay, so filtration of water and small solutes here, okay, they go through the gaps in the capillary walls. This allows water and nutrients to be delivered to the tissues, and it also removes waste from the capillaries in the kidneys. So let's look. You see the picture here. You have the water, the solutes, you have the capillary wall here, but then you see the cracks here, right? And the stuff and it seeps through and it's distributed. Here's the red blood cell. Now, it's not going to let the blood cell go through, but it will let the water and the solutes go through. Okay? Simple diffusion. Simple diffusion is the movement of particles from a high concentration to a low concentration. This is due to the constant spontaneous molecular. The molecules, they're busy bouncing off one another and they hit one another. A simple diffusion would be if you have a cup of water and you put sugar in it, it would eventually diffuse within itself. It would evenly distribute itself. Okay? Substances diffuse down their concentrated gradient. It does not require a membrane. Substances can diffuse through a membrane if the membrane is permeable to the substance. That means it'll go through if it allows it to go. Osmosis. Osmosis is the flow of water through a selectively permeable membrane. Okay, water moves from one side where water is more concentrated to be on the side where it is less concentrated. Okay, and then you have a solute particle that cannot pass through the membrane, so it draws water from the other side. A perfect example would be IV fluids. Now, when there is an imbalance in this, and then like say in the human body, diarrhea, constipation can occur, and edema. And if you guys don't know what edema is, you will learn, but I'll tell you now. Edema is the swelling or the fluid buildup in a particular part of your body. Normally, most people get edema in their feet. Tonicity. First of all, let's talk about, this is osmolarity and tonicity. Okay? Tonicity is the ability of a surrounding solution, like let's say a bath, to affect fluid volume and pressure in the cell. Depends on the concentration of the non-permeating solute. Okay? So we have a hypotonic solution. It causes the cell to absorb the water and to swell. That would be this little guy right here. It has a lower concentration of non-permeating solute than intracellular fluid. Distilled water is a perfect example of this. Okay? So this is your hypotonic cell, and that's what happened. Okay? You have hyper with the ER, hypertonic solution. This causes the cell to lose water and shrivel up. Crenate, it has a higher concentration of non-permeating solute than the intracellular fluid, and that would be this cell right here. It's shriveling up, it's shrinking. Then you have an isotonic solution. This causes no change in the cell volume. It stays the same. An example of this, when we get dehydrated, we start looking like this. Well, if we're given normal saline, 0.9% of normal saline, we'll start looking like this guy, a normal cell. Carrier-mediated transport. Primary active transport. The carrier moves the solute through the membrane up its concentrated gradient. The carrier protein uses ATP for energy. Examples would be the sodium potassium pump, and the calcium pump. Vesicular transport. It's a mechanism for exchange of proteins and lipids between the membrane organelles in eukaryotic cells. What it does is it moves large particles, fluid droplets, or molecules once it's through the membrane in a vesicle. A vesicle is a bubble-like enclosure. I'm going to explain how this works. Endocytosis. That is the process of which cells take in substance from the outside of the cell by engulfing them in a vesicle. So let's say that the material is near the cell. What the cell will do now is it will enclose it, seal it, break off within the cell, bringing it into the cell. Then you have phagocytosis, which is cell eating, engulfing large particles. Pseudopods are finger-like projections that take in the large particles. Macrophages, that is a type of white blood cell. What it does is surrounds and it kills microorganisms and removes dead cells. It also stimulates the actions of other immune cells. Now we have phagocytosis, which is cell drinking. It's taking in extra fluid, okay? It's the ingestion of fluid by the cell. By the membranes, they have these little budding small vesicles on the cell membrane that takes in the fluid that the cell needs. Exocytosis. Exocytosis is just removing, taking away material that the cell does not need. It's removing it from the cell. Vesicular transport, here's the process of exocytosis, that is secreting the material, the replacement of the plasma membrane removed by the endocytosis. And this is the end of part 1 of 2. I will put up part 2 shortly.